Good afternoon, young scientists and all the judges of Indonesian Science Project Olympiad 2021. My name is Daniela Putri Nugroho.
And my name is Clarissa Zefanya.
We're from 10th Senior High School of Bekasi.
And it's such a pleasure for us to join this competition. Our title project is
EFEG,
Eco Green Fertilizer Polybag. So without further ado,
let's get to the video! First of all, what does EFEG mean? EFEG is a polybag that is environmental friendly and more efficient than ordinary plastic polybags. From what we know, plastic is one of the biggest pollutants in the world. Synthetic polybag waste can emit dangerous methane gas and carbon dioxide, which is very toxic. As a result, these polybags are a threat to the fulfillment of food and human life. EFEG is made from the easily found ingredients, namely cassava skin and shrimp cells that were taken from a traditional market in the city of Bekasi. Research purpose. First, to know that EFEG is more environmental friendly than other polybags, and to test the effectiveness of EFEG as a planting medium and fertilizer for mustard plant. Second, to know the economic advantages of EFEG compared to other polybags.
Now, I'll explain to you the methodology of EFEG. As you can see, there are 4 different samples with different ratios but the same treatment. In sample A, we have the ratios of 3.4 to 1. In sample B, we have the ratios of 5 to 1. In sample C, we have the ratios of 9.3 to 1. And in sample D, we have the ratios of 75 to 0. And all of these 4 samples will be mixed with the same amount ratios of glutinous rice flour and rice flours of 20 to 80. Next, I'll explain to you how to make these EFEG polybags. First, you need to wash both the cassava skin and shrimp shells with running water until they are clean. Second, you need to dry them under the sun for over 1 day. Third, after drying, the cassava skin and the shrimp shell will be meshed and sheathed using 40 mesh sheath. After that, you need to mix both the cassava skin powder and also the shrimp shell powder. After the cassava skin powder and the shrimp shell powder already mixed, you can add warm water and the right amount of glutinous rice flour and rice flour ratios of 20 to 80 and make sure it was mixed well. Then you can put it into the mold and dry it under the sun for 4 days. Then your EFEG can be formed. Then we have 2 parameters. The first one is physical parameter. Physical parameter includes the plant height, number of leaf, and leaf area. Second, we have chemistry parameter. Chemistry parameter include nitrogen, carbon, and water content. Nitrogen is used to help stimulate the vegetative growth, especially in roots, stems, and also leaf. Nitrogen is also helped to make chlorophyll. Carbon is used to form the body of the plants for their physical growth. And water content is needed to help accelerate metabolism, especially in photosynthesis. And water also helps to carry out the photosynthetic product from leaf to all of the parts of the plant.
Research result. Observation result of EFEG application on mustard plant height. As you can see in the table, plant B has the biggest impact, which has 5.95 cm on the final day. And sample D has the smallest effectiveness, which have 3.26 cm on the final day. This is the graph of sample A, B, C, and D. As you can see, sample A and sample B plant height has increased. Meanwhile, sample C and sample D experienced drop. Result of observation of EFEG application on the number of leaves of mustard plants. As you can see, the sample B has the greatest impact again. The sample B shows that it has the greatest number of leaves on the final day, and sample C and sample D has experienced drop again. Why does the sample B has the greatest impact? The sample B nutrients and elements is balanced and the needs is fulfilled.
So, this is the graph. As you can see that sample B has the greatest effectiveness, which has total of 6 leaves on day 7, while sample D has the smallest effectiveness, which only has total of 2 leaves on day 7. EFEG application observation result against leaf area of mustard plants. As you can see, based on this table, that sample B has the greatest effectiveness than the other sample. It has 3.80 cm square on leaf area, while the sample D has the smallest effectiveness with only have 2.17 cm square in leaf area. So this is the graph that shown that sample B has increased on leaf area while sample D have decreased. Conclusion: we can conclude that with the existence of EFEG polybag, which are more environmentally friendly than the other polybag we use in general for agriculture and cultivation section, because the other polybags are used from factory produce waste and household processing, which will disrupt the surrounding ecosystem if it's thrown away without recycling it first.
Thank you for watching our video!